good. I know. Thank you. So hello. My name is Alexandra Ivanov. I'm a biology major here at Boston College. And this summer I've had the pleasure of working in the Naughton Lab researching bioelectronics, measuring and manipulating membrane potential. So the objective of our research is to understand if and how one can use membrane potential as a gauge of electrical properties of cells in order to study pluripotency, which is the capability of stem cells to differentiate into any cell in the mammalian body, and to also study differentiation for the potential use in regenerative medicine. So today, I will provide some background on the significance of membrane potential, how we are measuring and manipulating membrane potential, the fabrication of the sharp tip electrodes that we're using to do this, and then how we are understanding the relationship between pluripotency and membrane potential. So it turns out that different cell types can be distinguished based on their membrane potential. So for example, looking at this image, we can see that highly proliferative cells, such as embryonic stem cells, have a membrane potential of around -10 millivolts, while cells such as neurons can be characterized by a membrane potential around -70, -80 millivolts. So what is membrane potential? Well, it is the difference in electric potential between the exterior and interior of a cell, or in other words, the difference between sodium and potassium ion concentration across a cell's membrane. This then begs the question that our research will try to answer. Can we manipulate cells by changing their membrane potential? In recent years, studies have actually come out showing that changing bioelectric activity can affect functional regeneration of cells and tissues. In a 2019 study alone, they actually applied an external electric stimulation on stem cells in order to stimulate proliferation and differentiation. However, what these authors were unable to conclude was that they weren't able to connect how this external stimulation affected membrane potential due to the limitations of using voltage-specific dyes. Thus, there is a need for a direct way to measure and manipulate membrane potential, which is where our research comes in. So the first goal of our research is to use a sharp tip electrode to measure membrane potential, and then the second goal being to use this electrode to manipulate potential. So we would do this by inserting an electrode into a cell and either applying a voltage or getting a voltage reading. Now, some advantages of using a sharp tip electrode compared to the current methods of measuring membrane potential, which is the patch clamp device, is that our method is actually much less intrusive and cells can actually survive longer with it. As for long-term goals, we would want to extend this to coaxial arrays, meaning instead of just using a single tip on one cell, we could use this array to interact with many cells. So potentially, we could differentiate stem cells into a whole multitude of, let's say, kidney cells, all at once. So, thanks to the Zelkovich lab, we can reliably make these probes on the order of 100 nanometers. Now, for reference, a human hair is 100 microns, so our tips are 1,000 times smaller than a human hair, which is insane. And so here is an image that was taken on a scanning electron microscope, which actually allows us to see things on the nanoscale. And we can see just how sharp and small these tips are. And if we zoom in, just by looking at the 1 micron scale bar, you can see really, really small things. So, as for the fabrication, we start off with a tungsten wire, then use electrochemical etching to create the sharp tip. But in order to measure voltage, we have to first insulate this wire. So we deposit a chromium adhesion layer using e-beam evaporation. And this actually helps the insulator adhere to the tungsten wire. Then we can move forward with insulating using silicon dioxide using E-beam evaporation again, or with alumina using atomic layer deposition. But then in order to actually get a voltage reading, we have to expose the metal tip. So we use a focused ion beam mill to expose the tungsten tip. And here is another SEM image of our tip after insulation and the focused ion beam milling. And what we can see here is a much lighter exterior, which is our silicon dioxide insulator, now in orange, encapsulating a much darker core, which is our tungsten wire, now in green. And what we can observe from this image is that silicon dioxide has been effectively deposited onto our tip. However, in order to definitively ensure that our tip has been insulated, we measured current as the tip was incrementally dipped into phosphate buffered saline, and then did the same thing using an uninsulated tip to compare the two. So in this graph, which is plotted distance submerged versus current, we can see that for the uninsulated tip, as more area is exposed to the solution, so as the tip is dipped deeper and deeper into the solution, current actually increases. Now what we would expect for the insulated tip is for, once the tip to be immersed, current to stop increasing. However, what we observed is a slight increase in current, meaning that the tip may not be fully insulated since current is still leaking through. This has caused us to move away from using silicon dioxide as our insulator and instead moving towards using alumina, since a preliminary test of it has showed, a preliminary test of measuring current as we dip the tip into phosphate buffered saline has showed that current was 0, which means that our tip was effectively insulated. So as we move forward with our experiments, we will be moving forward with alumina as our insulator. So now that I've talked about the fabrication of our sharp tip electrodes to measure and manipulate membrane potential, I'd like to talk about the bio side of things, which is how we explore the relationship between pluripotency and membrane potential. So it turns out that pluripotent stem cells keep their intracellular potassium concentrations anomalously low. So why is this? Well, there is this microRNA, miRNA 367, which is a non-coding RNA that acts as a silencing molecule to modulate gene expression. Now this miRNA is essential in pluripotent stem cells and actually maintains pluripotency. Most importantly, it is regulated by intracellular potassium concentration. So what does this mean? It means that when potassium concentration is high in the cell, the pre-mRNA structure will equilibrate to the G-quadruplex structure, preventing effective dicer enzyme processing, and thus inhibiting pre-mRNA maturation, which is so essential to pluripotency. So, we hypothesized, will stem cells remain pluripotent if excess potassium is added to their media? So we cultured our stem cells in 100 millimolar and 0 millimolar potassium gluconate solutions, with 0 millimolar serving as our reference. And we expected that 100 millimolar would be too high for the sodium-potassium pump to compensate, and thus for an increase in intracellular potassium concentration to occur in these stem cells. And we then visualized potassium ion movement using Asante green, which is a potassium ion specific dye. And what we can observe in this image is that in 100 millimolar potassium gluconate, the stem cells actually had an increase in fluorescence, meaning that there was an increase in intracellular potassium concentration. But how do we know then if these stem cells actually remained pluripotent or lost pluripotency? Well, there are these two transcription factors, and transcription factors regulate gene expression. So these two, SOX2 and OCT4, are essential to pluripotency, meaning when SOX2 and OCT4 are no longer present in a stem cell, these stem cells are no longer pluripotent. So we stain these stem cells with red fluorescent antibodies to SOX2 and OCT4. And then also stain DNA with blue fluorescence in order to just guarantee the presence of our cells. And again, we use 0 millimolar potassium gluconate as our reference. And we observed our stem cells in 100 millimolar potassium gluconate 24 and 72 hours after staining. And what we can see after 72 hours is that red fluorescence no longer remains, meaning that the transcription factors are no longer present in these stem cells, and thus, these stem cells have lost pluripotency. Now, what these findings suggest is that an increase in intracellular potassium concentration in stem cells leads to a loss in pluripotency, which would open the doors to countless possibilities in the field of bioelectronics. So, I would just like to thank Dr. Naughton, my PI, as well as Dr. Connolly for all of his patience with me, as well as everyone in the Naughton lab. Thank you so much for supporting me. Specifically my mentor Mark for putting up with a bio major in the physics lab, and my wonderful lab partner Megan Macy, as well as Hong Lee for his such great help with the electrochemical etching. He did not have to do that, but he always helped me. And of course the VCRU program, and all of you for being such a wonderful audience. Thank you.
Any questions?
Anyone? Yes.
Do you only look at cells that are pluripotent, or do you take in cells that have different degrees of pluripotency?
We actually use a media that selects for pluripotent stem cells, so we only use cells that are pluripotent in at least these studies. Anyone else?
Okay. So that will conclude our presentations for today. So look forward to seeing you all at the banquet at 6. Thank you.
That was the very last one.
I love that you guys
Just like generally telling science stuff that actually goes quicker rather than I mean, inherently, yeah. But I might like force them to be losing the ground.
Where are you guys going?
Yeah.